In the deep ocean, an eye may wait through darkness for a single traveling photon. Most moments deliver almost no image, only scattered light and the animal's own neural noise. Then one blue flash appears, and that nearly empty visual field becomes urgent information. The paradox is severe. Deep-sea eyes receive little light, yet must detect events that matter immediately. Some eyes grow enormous, while others shrink or disappear where vision offers limited returns. Between those extremes lies a collection of adaptations tuned to rare signals, not rich scenery. The investigation begins with a predator suspended beneath the last sunlight, scanning an apparently vacant world. Its retina cannot reveal everything because sensitivity, resolution, speed, and energy impose competing costs. Catch faint light. Visual systems often pool signals across space or time. Pooling improves detection but blurs detail and slows the recognition of rapid change. The eye, therefore, sees almost nothing by human standards, while extracting nearly everything necessary for survival. If you entered this darkness unaided, the absence of landmarks would feel complete. For adapted animals, that absence contains gradients, flashes, silhouettes, and rhythms. We will follow one diminishing light field and ask which information remains trustworthy. The answer depends on depth, habitat, behavior, and the kind of photon an animal needs. Sunlight weakens sharply with depth as water absorbs and scatters its wavelengths. Blue light usually travels farthest in clear ocean water, shaping the spectral window below. By the mesopelagic zone, downwelling light remains detectable, but may be too faint for color-rich scenes. Deeper still, sunlight disappears, and bioluminescence becomes the dominant visible source. Most marine bioluminescence is blue or blue-green wavelengths transmitted efficiently through seawater. Many deep-sea visual pigments are correspondingly sensitive within that region. Large pupils capture more photons, and large lenses can focus more light onto the retina. Tubular eyes in some midwater fishes point upward toward silhouettes and luminous prey. Reflective layers can send unabsorbed photons through receptors again, increasing capture probability. Densely packed, rod-like photoreceptors favor sensitivity where color discrimination or fine detail may be less useful. The cue is faint, downwelling contrast. The action is upward fixation, and the consequence is earlier prey detection. This sequence is supported by eye orientation, retinal design, and observed feeding ecology in several fishes. Still, anatomy predicts capacity. It does not record exactly what an animal experiences. The barreleye fish Macropinna microstoma carries tubular eyes within a transparent, fluid-filled shield. Observations show those eyes can rotate, supporting upward searching and forward orientation during feeding. The cue is prey position, the action is eye rotation, and the consequence is maintained visual tracking. That capability corrects older interpretations based mainly on damaged specimens brought to the surface. Other fishes divide retinal fields or use accessory structures to monitor different directions. Some eyes emphasize the bright ceiling, while others emphasize flashes arriving from the side. The diversity reflects multiple darkness problems, rather than a universal deep-sea design. Verified structures show what light can be captured, but neural processing determines what becomes a signal. Photoreceptors must distinguish genuine photons from spontaneous molecular events and electrical noise. The evidence now sharpens around the smallest possible visual event, one brief flash. A bioluminescent pulse may identify prey, expose a moving body, warn of disturbance, or signal a mate. Sensitivity to such pulses depends on wavelength, duration, distance, and water clarity. Longer integration gathers more photons, but it can smear a fast-moving source across the retina. Spatial summation combines neighboring receptors, improving detection while sacrificing precise location. The cue is a weak flash, the action is neural summation, and the consequence is detection without a detailed image. This trade-off is mainstream sensory physiology, though settings differ among untested deep-sea species. Some animals possess multiple visual pigments that may separate kinds of blue light or broaden sensitivity. Several dragonfishes show unusual visual specializations associated with far-red bioluminescence. Malacosteus niger can generate far-red light and has adaptations supporting sensitivity beyond the usual deep-sea blue. This system may illuminate prey that cannot detect the same wavelengths, functioning like a restricted searchlight. The cue is nearby prey, the action is far-red emission, and the consequence may be asymmetric detection. Laboratory measurements support the optical capability, but natural hunting outcomes are difficult to observe. Color vision claims require caution because multiple pigments do not automatically prove behavioral color discrimination. Researchers examine anatomy, measure spectral sensitivity, record electrical responses, and observe behavior when possible. Each method answers a different question and leaves subjective visual experience unknown. Remotely operated vehicles provide behavior in context, yet their lamps can overwhelm natural adaptation. Animals may freeze, flee, orient, or alter emission because the instrument changes the scene. Low-light cameras reduce disturbance, but still struggle with the photons the animals themselves barely receive. Specimens recovered through pressure and temperature changes may show altered retinal performance. These constraints make strong conclusions local. A measured receptor responds, an eye rotates, or a behavior follows. They do not justify claiming that every deep-sea animal sees an equally vivid hidden universe. In fact, many deep habitats favor reduced eyes or reliance on non-visual senses. Deep-sea worms, crustaceans, and fishes can combine vision with smell, touch, flow detection, or vibration sensing. The meaning of almost everything is therefore ecological relevance, not complete environmental representation. The boundary of darkness, vision guides daily movement through layers of changing risk. Many midwater animals descend during daylight and rise towards surface food after sunset. Their eyes track fading overhead brightness, helping time migration without a detailed horizon. The cue is changing light intensity. The action is vertical movement, and the consequence is altered feeding and predation risk. Predators below search for silhouettes, while prey above watch for approaching flashes or shadows. An upward tubular eye concentrates resources on the direction where contrast remains strongest. A downward-facing visual field may inspect counter-illuminating prey or luminous disturbances below. When attacked, some animals release glowing material that distracts a pursuer from the escaping body. The cue is contact, the action is luminous discharge, and the consequence can be a diverted attack. Eyes sensitive to rapid spatial change help both attacker and target interpret that sudden light. Finding mates presents another challenge because populations can be sparse across enormous volumes. Species-specific light patterns may carry identity, though functions require behavioral evidence rather than visual resemblance alone. An eye tuned to a narrow spectral band can reject irrelevant wavelengths and emphasize familiar emissions. Yet narrow tuning risks missing unusual prey or signals outside that band. Energy also matters because retinal tissue and neural processing are metabolically costly. Evolution does not maximize sight absolutely. It balances useful information against construction and operating costs. If you examine a giant deep-sea eye, size alone tells only the beginning of that balance. Its orientation, receptor arrangement, pigment sensitivity, and neural wiring complete the functional hypothesis. Human observers often mistake bright vehicle footage for the animal's normal visual world. In reality, white illumination introduces wavelengths, contrasts, and ranges that may never occur naturally. The human stake is understanding perception without replacing it with our own illuminated assumptions. Future investigations need behavior recorded under natural spectra and intensities at depth. Pressure-retaining equipment could connect retinal physiology with undisturbed body condition. More species require mapped visual pigments paired with tests of actual behavioral discrimination. Neural recordings could reveal integration times and spatial pooling, though such measurements remain technically difficult. Free-swimming observations should compare responses to controlled flashes, varying in wavelength and duration. Environmental light measurements must accompany those trials, because sensitivity only matters relative to available photons. Researchers also need predator and prey visual models built from measured receptors, not human camera output. Comparative studies could test whether eye enlargement tracks depth, bioluminescence, body size, or hunting strategy. Reduced eyes deserve equal attention because they identify conditions where vision no longer repays its cost. Unknown neural processing remains the largest gap between receptor measurements and perceived scenes. You can return now to the waiting predator beneath the last faint ceiling. For long seconds, its retina may register no stable object and no textured landscape. Then a blue pulse crosses one sensitive field, separating biological event from background noise. The eye may not resolve the source's full shape, color, distance, or intention. It extracts direction, timing, brightness, and motion, enough to trigger approach or escape. Almost nothing becomes everything, only because survival values a few photons more than a complete picture. Deep-sea eyes do not conquer darkness. They conserve its rarest information with measurable, imperfect precision.